responsive team, which has led to this development that we are presenting today. So, as Lucilla pointed out, we use the acronym DATS as an echo to the format that is used by the publisher to deposit information about journals to the PubMed, and so that it can be indexed and searched by the PubMed engine. So really the DATS could be viewed as a DATS is needed for a scalable way to index the information. So the work that we are presenting today has been published and released in various forms. The most present we've been using GitHub to do all the development, and every release point has been available and made available and DOI through the Nodo repository. Google Docs have been used for collaborative development information. And as you can see, a couple of publications have resulted from this work. We've one that has been published in Nature Genetics earlier this year that summarizes the main deliverable, as well as the more technical information, which is published in Scientific Data that gives you an overview of the model, the detail of the development. And of course, all the slides are available on our GitHub repository to describe the metadata standard. This is a kind of overview of the timeline from the moment we started about 2 years ago now in March 2015, when the first use case workshops were carried out, until now, basically in May 2017, with the latest release of the BioCaddy DATS model 2.2, which incorporated a number of feedback that we received during the BioCaddy CDE meeting in Bethesda earlier this year. So really, in August 2015, we released the first version of this model here, and that was through a set of conferences and iterative process of development, consulting a number of use cases. This led to refinement, and in May 2016, we had the first release of version 1.2, and then a month later, during the first DATS workshop, which was held last year in San Diego, we carried out a kind of first attempt of socialization of the model to see how people from different repositories would interact with it, and how the kind of feedback we could receive, because this is again a very live interaction model where we need to provide to obtain input from people meant to implement some of these elements. So really, the model is not meant to be a perfect model that solves all the problems, the modeling problems of the life science domain. Rather, it is by essence some kind of simplification. We had to make compromise because this is the nature of the work. We need to extract information from a wide range of repositories, which are specialized resources with specific data models. At the same time, we have to frame this activity, which is really at the core is the findability and accessibility. We need to make this information available, and just refocusing on the first two letters of the FAIR acronym. And really, one of the constraints we had was to somehow, based on the use case that were provided to us, try to maximize the solutions that we could afford within the envelope of time allocated. So the development process took two facets, essentially. One was drilling into a number of use cases which have been provided to us. Some of them are very granular, as you can see here. So for instance, search the study for patient data with biological process X, for instance, obesity as measured by BMI, and a set of interventions, and then filter of demographic characteristics. This use case means that we have access to a number of patient characteristics, and immediately it runs into a problem of possibly privacy issues related to human data. So then brings up immediately the notion of, or key concern of, how do we summarize the information about accessibility of the data set and permissions, condition of use, so that people can understand, and this information can be surfaced from DataMed, allowing them to there drill a bit deeper into the data set itself. The other side of the approach is that as we were meant to be interfacing with a number of repositories, we had to understand the kind of data model they themselves were using. And basically, we used the biosharing catalog of models and standards that is available in our team to get an insight into the kind of metadata that was actually used by repositories such as Geo or Express or Metabolites, for instance. And that mapping work led to the development of the DATS model. So this gives you the list of the core entities that are actually underlying the DATS model, and this resulted from the convergence of the set of competency questions that were provided to us by these working groups, which had been gathering use cases from scientists, and as well from the mapping of about 30 models that are actually used in the wild. And so there are two parts. As you can see, there is one core that really characterizes the main feature of the data sets, and then an extension which somehow focuses more in drilling into more details that are specific to certain type of data sets, mainly related to studies, proper studies where you've got an intervention, which might not be the case in other cases. So therefore, we had this need to provide a kind of adjustable envelope for metadata to suit the various data sets from different sources. In terms of adoption, what is interesting to notice is that as we were working with various groups from the Force 11 project, we were also working with people on the DATS side, the project behind DOIs, which also have a number of needs to store descriptors for metadata. So in terms of mapping, I would like to drill a little bit into some of the models that we have investigated. So going to the biology side and the kind of major repositories that we are aware of, such the one that are hosted by the EMBL-EBI or the NCBI here in the US, we obviously had to look at models such as the BioProject, BioSample, but also the minimal XSD format, which is used by Geo for microarray data, proteomics data with MHML, the GH4GH metadata format, again, to understand the kind of information exchange that is needed in that realm. And of course, we also looked at the ITAR model, which is kind of a synthesis of some of these model that I just mentioned. On the clinical aspect, we looked at the CDISC model, but also the OMAP project. I will mention that a bit later at the end of my presentation. And then we looked at more hardcore, I would say, or focused metadata projects, which are the one that from the data side I just mentioned, the REEF-CS or the HLS dataset initiative from the W3C consortium, which had been running for a couple of years before, for which we had to look into these aspects. From an early stage, we also looked at schema.org. Again, we were aware of this project in the world of indexing, and we thought it would be a good idea to poke it to see how far it was supporting the use cases that we were confronted with. And so there was a schema.org initiative, but also there was schema.org supports extensions to that. And one of them, the life science extension, had a kind of significant vocabulary, but it was very restricted around clinical trial, which meant that a number of cases that were supposed to be supported by DATS and DataMed were somehow out of the window here. So again, following this mapping and this landscape survey, we thought, okay, we have enough material to understand the capabilities of some of the resources and the choices that we need to make. So really, as I said, we have a kind of two-tier model, DATS for the core and the extension version. The core is meant to be applied to any kind of data source, and therefore provide the objects which are required to provide a big description, a major description of the key features, like the title, where to find it, how to access it. I will get back to that. And then the key point is that all these entities are not necessarily mandatory per se. We don't force anything to be used. This could be delegated to a metadata profile that could be checked later on. And therefore, these are the kind of, we didn't want to overburden the users and the implementers. At the same time, when we move towards production, we need to ensure that the quality, the data meets a number of quality requirements. But that's, we don't want to constrain too much the model at this stage. So also we had to distinguish between two types of data that we were still discussing whether to index that in DataMed or not. But we had to distinguish reference data sets or data from knowledge bases. For instance, the set of protein as identified by UniProt, whether we want to index this in DATS or not. But also, and this differs from data sets where signal is actually measured on a population and may need the information from the previous kind of data set that I just mentioned to understand how this relates. So this information about studies that as we can find them in dbGaP or GEO or clinicaltrial.gov, again, allow the definition of population on a number of inclusion criteria which need to be evaluated or surfaced in DataMed, and how we capture this information is essential. So this is a busy picture of the model. So I won't have time to drill too much into this, but as you can see it again visually, the gray shaded boxes are the core elements, while the orange ones are the extensions which allow support of a very fine or finer grain description of a data set of such the one that we find in clinicaltrial.gov, for instance. So these elements allowed you to basically identify or mark up the key facets of a data set. So what has been used to produce signal in the data set. So we need to focus on the kind of material or data. Should it be an in silico study? And how you produce this signal. So we need to be able to understand the kind of dimensions, the variables, essentially, that have been measured or used to filter a population, the kind of methods and technology that's been used to acquire the signal. These are main classifier or that allow to slice and dice the information as you can discover it and explore it in DataMed. And key to the information, as was touched upon earlier on by Jeff, is you need to be aware of how and what you can do with these data sets once you've identified it. So working with George Alter and his team and many others in the group, a specific extension and modeling has been performed to really make sure that the information about the condition of access, accessibility rights, format and distributions of the data set was surfaced. And this is important in the sense that we wanted to make sure that the information available from DataMed could be available programmatically, as shown by Hua earlier on through a web API, and possibly channeled to analysis tool once the filtering had been refined. But to do that, we need to understand the protocol of access and the formats available. So then, of course, we need to keep a tab on who did what and when. So this is about the notion of traceability and citability of a data set to provide the means to somehow gauge the impact a data set can have on scientific output. So again, these are maybe seen as kind of housekeeping metadata, not necessarily drilling into the meat of the data set. However, in the long run, these are essential, critical metadata to understand the usage and track traceability and impact of a data set, as well as the information about the organization, the persons, and the grant that are supporting these efforts. So again, another picture, but a bit more color here, where we can point to the by different color coding. So in purple, you've got organization, persons, and materials. In green, we've got the kind of information and about ground, places, dates. Actually, I can point out that these elements here, place, geospatial coverage, and dates, were additions following the first feedback that we have with the user following the initial meetings. That, again, provided a key information about what was missing from the elements. So now I would like to switch gear and to mention some of the work that we've been doing around schema.org and as a kind of test finding. So as I was mentioning at the beginning of the work with DATS, we looked at the various models, and we obviously looked at schema.org owing to its origin and its use by search engines, and we had to keep an eye on that. So as DATS is entirely developed as a JSON schema, we thought it was a very valuable exercise to develop the JSON LD context file using schema.org to showcase the alignment and possibly submit requests to the schema.org effort to support a direct markup of pages in repositories using a DATS profile under schema.org. And this is what we just have done. And I will point out that this is also a coordination with an effort in Europe, which is called Bioschema, which focuses itself on working on specific profiles to identify and model biological entities or entities relevant to biosciences, which are currently missing from schema.org. And just to touch briefly on to show you how the impact that BioCAD has had already, this is a page directly posted from Google development pages and showing the dataset profile, and I've highlighted in red the attribute which is called viable measured. They couldn't use dimension, which is coming from that, but this is the same concept basically, and it's mapped directly into that. And recently, just last week, as I was spending more time in San Diego, attending the 4th11 Scholarly Communication Institute summer school, talking with David Kennedy, who runs a large repository of fMRI data, we were discussing that this is not enough, and we would need to also have another attribute to identify the kind of methodology and technology type to slice again and be able to refine the searches on the repositories. So again, it shows a path on how BioCAD and DATS can interact with an effort such as schema.org to channel requests, and again, work in a push and pull work. We can work actively harvesting a lot of data from the repositories, but we could also work towards helping them mark up their pages with a profile that would be aligned with DATS, and we could automatically harvest using robots to scan their page or web services, and therefore make the life of indexing smoother, but also the adoption of the work that has been done by the BioCAD project seamless and easier on those repositories, owing to the pool that's interacting with a search engine and having the ability to search directly in these pages could have. I won't go deeper because there is an update on this project on the afternoon, but this is again a last bit that I would like to talk about the data standards and how also this is important to track the quality of the metadata that we could extract from repositories or as advertised by repositories. It's a link to the data standards and the specific relation that the dataset distribution conforms to a specific standard. And basically, again, we can relate and link up to the biosharing or fair sharing effort by providing unique identifiers to these elements and again, make the data consumable by automated engines. So in the last few minutes, I'd like to talk about some of the DATS implementation that we have been experimenting with, and this is the result of the Bethesda meeting that we had earlier on in this year in May. And we've been working with three major repositories. One is Import, the Immunology Database Analysis Portal, which provides a big repository warehouse for a number of clinical trials around, looking at the immune response in a various contexts, and we've been working with them so that they could work on an export directly from DATS. And this interaction revealed that we had a good mapping However, we could have improved, or one of the struggles ahead was probably to have a better guidance. We were lacking documentation on how best to represent the variables and the dimensions that are actually represented, not only representing those elements, but also how to push the results in a more structured fashion. The other effort with the PSR, led by George Alta, which is an inter-university consortium for the political and social research. And again, they've used a specific format, the DDI, and where basically they can also store a huge array of variables that are critical to their studies and measure that in accurate detail. So really this is again pointing to the same direction. Once we have identified or provided the key metadata to somehow describe and understand how a data set can be available, we also need to open up to be able to drill into more detail. And again, this is not entirely obvious when we started this project, especially when we look back at the list of use cases that we had, which were in the hundreds basically. It was unclear where we had to place the cursor and how far we had to dig and represent the granularity of the data set. Now that we have provided the bulk and the framework, we started to get, okay, I found something interesting, now I want to delve into this data set, and this is the second layer of implementation that we probably need to go for, and it will probably be in the next 6 months the focus of refining some of these harvesting pipelines. The last one it was focusing on the observational medical outcome partnership, OMOP, Common Data Model, and this one is particularly interesting indeed, owing to its prevalence and the diffusion and uptake in projects such as Odyssey for tagging electronic health records and providing a huge wealth of information for human research. So again, this was pointing to the need for DATS group to provide more guidance and examples on how to represent this information about variables and what was the best way to do this. Also, specific to this model, the fact that there was the intuition that, or the impression that the DATS model was somehow focused on a study-centric element, and sometimes that was not so easy for people coding information in OMOP to represent that concept. So the lesson learned really was that we need to improve on how to make sure to identify the data set from particular sources and how this is done through the indexing. We have to document better how to use the dimensions and the different kind of dimensions and the different, whether it's quantitative or qualitative, and how to best surface this information so that it's not overburdening or overinflating the feed to DataMed. So there is a set of optimization that we need to look. Also, we had to improve our documentation by providing more metadata profiles and guidance to the users so that they can engage and pick up the skill faster. And then, of course, for the support in terms of infrastructure, that would also be important so that we can have guidance and a clear validation as well of the metadata profiles that could be sent to DataMed. Finally, I will finish on these two elements, focusing again on the refinement required to provide information within the DATS feed that will populate DataMed about how to provide and report dimensions. This is kind of close up on the model of dimension, how we can relate that to the different parts and how it relates to the data set, and how we can provide the values to this. The metadata profiles indicated on the right, and basically the next purpose will be also to provide an accurate mapping with schema.org and align, engage more with the community to see how that can contribute to this development. Give you here just an outcome, a profile of how things look like. Now that we need to focus on reporting also the value range, and this is an outcome of the work on the Bethesda meeting in May, where we had a very successful meeting, engaging with a number of repositories, NIH and otherwise, for understanding the common data elements which are currently stored and how those variables could be indexed in DataMed and possibly unified or at least mapped across so that the discovery of potential studies of relevance would be achieved at a more efficient manner. I think I will stop here and take any questions from you. Thank you for your attention.